Hey everyone, it's me again. Today, in this video, we're going to talk about how are you going to look at your PICS analysis. And after a long wait of your PICS analysis, now you're able to finally to see what is the fun things about proteins in your analysis. So I'm going to use one example I have here just to go over what you're going to see, a really brief overview of how can you look at the data. And as you know, we're doing proteomic, and there's thousands of data that you can look at. And then you really need to have the questions that you have as your guide to help you navigate through all this data. Okay, so today I'm going to use one set of voting data here. It is the set I'm going to use, and hopefully I have most of the data open up so we don't have to wait since there's enough analysis going on right now. So it might be slow it down if I need to open any of the tab. Okay, so let's start with what is mass spec analysis. What is mass spec really at all? Here is briefly we go through the chromatography data that you're going to look at. It has retention time on the x-axis and intensity. Usually it is displayed as a relative intensity here, and you can change it to absolute intensity, which is also listed here. So it is a 3D matrix. You have your retention times recorded, and you have your intensity, and you have your M over Z as a second metric, and you have your intensity. So, so that is all what mass spec will give you, and along with some other things your experiment acquisition method. But on the data side, it will give you these three metrics. So can you get protein ID from just these three? The answer is no. And if you have taken some organic chemistry, you're going in that class, you might need to put out a spectrum, analyze this spectrum by hand, and just looking at we're getting tens of thousands of spectrum in our analysis. You're not going to do it by hand, and so that's where protein identification software will come into the place to help you do those things for you. Okay. So this is today I'm going to go through voting data and just go into the couple steps of what we're going to see from the analysis. So here's the novel is the first step of the analysis, and I'm sure I did not open this one before, so it might take a while for us to wait for it to open up. So what it does is take all those three-dimensional data into the computer and calculate the theoretical the peptide ID for you, peptide sequence for you. So that's the first step. It tells you how many MS1 spectrum you have, how many MS2 spectrum you have, and then how many peptide sequence according to the calculation we have for mass spec. And it's still taking. I'm running out stuff to say. Yes, and I don't know how long it will take, but it is something that you know that if you run analysis, there's analysis open, running, it will take a while for you to open each tab to do the analysis. Okay, so I'm just going before we go into the tab. Let's just the novel is the first step. It takes analyze your three-dimensional metrics into convert it into peptide. It do all the calculation for you. And then the next step, actually the next three steps, is all about protein identification, the PICS database, PICS PTM, and SPIDER. It all does a little bit different things. So PICS database is the first step that here, this is the first step you're going, the mouse, we'll just follow the mouse because I cannot highlight it right now when it's running. Oh, finally got that. Okay. I'm just going through this PICS database tells you like match up like the peptide you have from the novel to a protein sequence database that you put in your like the species you select when you queue up the analysis. Match it with the protein and see if it matches, and PICS PTM look at the post translational modification on your peptide, and spider like look into point mutation. And we'll go over to these three in a bit later. And label free like do a relative quantification for you, like between all your sample sets. So if you have only one sample, you would not be able to do that. But if you have like two more samples, you can do that. Okay, so here is the de novo tab. It tells you like how many MS1 you have or how many MS2 you have. And looking at here, like we are dealing with like 100,000 of MS1 and then 450,000 of MS2. Okay, imagine if what like if you did the like spectral analysis trying to piece up the peptide or understand what it is, the component you have, like you would imagine like how long, how much time it would take. Well, I would say it's nearly impossible for me to analyze this set of set and do the protein identification. Okay, and after the de novo, we have like 130,000, 130 of thousands of peptide. So, and they also give you like your basic like experimental like instrument parameters and your PICS analysis setting. Okay, and here in the de novo tag, it tells you, yeah, like from which sample, like for this set of analysis, like this one, this analysis, I queue up 9 samples. So it tells you like which samples you have version 1. F1 is this sample. From that files, you have this feature ID, and we got a peptide here, and it is like 7 residue long. The score is like your confidence of how real it is, and M over Z, and the charge and the retention time it is, and delta retention time between samples and the area you have there, and the ppm, like ppm represent like ppm arrow to the like theoretical mass. Okay, so later on in biochemistry or like 583 if you're a biochem major, that you will learn about how you're going to use MS, like mass spec, to do the like protein ID. But the idea is we have a MS1 precursor that is our MS1 precursor, and we measure its M over Z once, and after that, like the instrument break it down into pieces using MS2, like just like framing it further, that like fragmented this precursor further. So that's our MS2. So we have all these small pieces there, like just again, just to show you a better idea of this is MS2 spectrum. So after our MS1, we select one precursor and one mass and fragment it into all these fragments that we have from that precursor. Okay, so coming back there, and this is a lot like a more clear spectrum here. So from there, like we have each pieces, and we can like calculate the mass of like Y ion and B ion, and we can calculate the differences between each pieces. And so because protein is made by peptide, and peptides made by amino acids, and we only have 20 amino acids that is common. So the delta mass between each ion peaks should be fixed. And that is how we are able to calculate use the differences between peaks, and then calculate the difference and find out, okay, which amino acid is that, and like then we can piece back together and make the peptide sequence there. Okay, cool. So the blue is, or the red, like blue is for B ions and Y is for Y ions. But the more you have, like it also calculates the add that we have. the more we like, the more color we have, like that means we are more confident to this peptide that is real. Okay. Just looking at like a 3D feature. So like that is de novo, and this is like feature ID. So if you look at like, you can sort by feature ID, and we have like 50,000 of it, 50,000 features. And then you can look at it and see how many like spectrum we get from one feature. Okay, so this is healthy MS tab. Come on. Okay, here we can see this is a 3 dimension, like M over C, and sorry, retention time M over C, but you cannot really see your like you have your intensity on here, but you can use this bottom and make it 3D. So you can see like how that is 3D. I really like this. Okay. So the following tabs that I'm going to show you have like similar features of each thing. So let's see. I did not open this tab also. So here is a brief explanation of like how you can look at some of the data, and you will learn from your experiment, your data, like to be more familiar with peaks and freestyle, because you're going to have more questions to ask, and that is how you're going to be proficient on the software. I honestly speaking, there's a lot of function on this software I still don't know. So it's a learning curve. And then the good thing is, if you ever have questions that you could not find any grad student on Sense that know the answer, you can always call Peak Studio. They are really responsive on answering your questions. Like they're really good. Like, and actually we pay a lot for like their license to license their software, and they did really good at answering your questions. So like asking question is crucial, and asking the people that have the answer is learning to ask the people that has the answer is also good technique. So call them, and then the check is like really friendly, really responsive. Okay, so if you have any questions, then you can ask them. And then let's see. Well, I have done talking, and it's still not pulling out. Let's wait a bit. I'm going to pause it for. Okay, now I resume the analysis, like the videos, and it is opening up Peaks database. Okay, so the following three tabs: Peaks database, Peaks TDM, and Peaks Spider have really similar, like pretty much exactly the same output. So first, here you're going to see like that is your false discover rate. And you want to see a pretty exponential curve like this, because sometimes you'll see something like going up and start like noising here, and then your number FDR is at 100%. So false discover rate is about like how like telling you, and similar, it tells you how accurate your peptide identification is, and also your like yes, and then your peptide, and you're like how well it is matching the database, protein sequence database. So you want it to be high, and you can set the FDR here, and it is default to do like negative 10 log P. So it depends on that, it might change your FDR, but like you can see, like most of your peptide have like 28,000 of peptide are below like 0.5%, and then most of them are below 5%, which is really good. So here, and you also like it tells you a decoy and the target, and you want like most of your stuff are blue. Here is like your target and your mass arrow, and you really want your like your peptide are the blue. You want it pretty much within like plus and minus 5. So it's like this set is not bad, but we do know it has a 10% ppm shift. Okay, so here, like I said, like this is a nice set of samples. It tells you how many, like for each one, how many MS1 you have, how many MS2 you have. Here, like just looking at like for the nice samples, like we are looking at like a million of features. So like just like I say, like this set of samples take about like more than 24 hours to finish the analysis, or about like 18-ish, a long time. So like the more samples you run, just be prepared. You link to have more time, and also like this set of samples have some PTM I'm looking for specifically. Okay. Also, I hear like every time you want to look at how many peptide and how many protein you have, notice that like this is like the protein like total protein. I only have 400 like total across the samples, and which is weird. Like if you ever run monthly, for example, like you're looking at like 2,800 to 3,400 ish. That will be your standard. And if you're looking at my data, you will feel like, oh crap, like what did I do wrong? But this is actually good for me, like not too bad for me, because I'm actually running a serum samples, and we know serum has a dynamic issue. So it is like that is like low protein identification is a common problem with unfractionated serum, and I run it unfractionated. Okay, so if you have less than 8 samples, it actually will add, like if you have less than 3 samples or 4 samples, it will give you a Venn diagram, tell you like what portion, like the overlap of like the protein and peptide ID, and if like there's any unique protein for each fractions. And if it is like less than 8 samples, it will give you a table to you to read that. But because I have 9 samples, it will not generate that for me. So that is a like brief analysis you get from here to get a brief feeling of your analysis, and it tells you like the distributions or histogram of your like MRZ and retention time. And so it is a 120 minutes run. So you can see I have a lot of stuff at the tail end. Okay, and the like distribution of your signal intensity. Okay, and at the end it tells you like the parameter of your settings. And so this is a database, like putting the database search. And also there are some of the other things that we can go in more detail later. But here's I would like to tell you. So the features tab is similar to what we see in the de novo. And then protein, here's what I'm going to show you how we got a protein identification. Okay, this is serum albumin. It's the most abundant protein in serum, so you're going to see a lot. So here, this line tells you what protein you have, is p-value, and we want like for like anything greater than like -10 log p20 is good. It tells you the protein coverage, so how much your protein is observed in your mass spec run. So what does mean for coverage? For this big protein, is hard to see because it's a huge protein. Let me find something that is smaller, like this one. Okay, I don't know what this protein is. It's CD5 something, and you get like you can know the description of it. It's an antigen. Yeah, so this is the protein sequence. It has about like 300, 20, 30, 40, like 340 residue. So all this is the knowing like protein sequence that has like the protein that has been sequenced, and when they update the database. So the blue peptide is the observed peptide that has the like p-value greater than 20. Like this one, it only has a score. It doesn't have p-value. The blue one is the one that has good, like you observe and has good p-value, and the green one is you observe that doesn't have good great value, so it will be excluded. So you can see, so for this protein, you can see like about like 51% of the sequence is covered. We observe that, and those like this region, we don't observe it. So that's like what is mean for protein coverage. And we see like 17 peptides and 17 peptides are unique to this protein. So we know like protein has some hormones, but like for if you're looking at, let's see, some immunoglobulin, and you can search your protein by here and changing like your like accession number or in immuno. Okay. Hope I can spell. Like looking at here, this is one IgG, one immunoglobulin, and you observe 117 peptide, but only 48 is unique to this protein. The other is shared with other like all the other like 50-ish, 60-ish are shared with other proteins. Okay, so this is I tells you like what the like coverage, and it tells you like for the coverage of like the protein coverage for each fractions. And like if you go descriptions, and there's a PTM tag here that tells you what PTM we have observed. If you have observed one PTM, you will show it up here. Like I have a box on your peptide sequence, and you can reference it about to see what it is. Okay, and like you can, that's for protein, and for you want more detail for like this protein, particularly like for the peptide, you have peptide sequence, p-value, mass, length, ppm, everything, and like the area of each fractions, and you can get all this like exported out. So you can go here and to export and export text format, that would be good, and just to the export folder. I'm not doing it now. Like, and we're going to have another video explain like the export of Excel later. But so that is like how you're going to look at your protein. And so just and for this particular experiment, and I like to see my transferring, and I have been working with it a lot. I know the protein accession number. Search for it, it will bring it up for you. So that is it comes through. There's so many data. What are you going to learn? Like how are you going to learn it? That is why like your objective in your project is important for each experiment, because that guide you to know like what you're going to learn. So for this project, I'm working on an EDTA experiment that will chelate out the iron from the transferring. So transferring is what I'm interested in. So I come here and search it for first. Okay, so I bring it up and it tells you like it have like good p-value, 79%, and then all of them are unique for itself. Okay, just a couple of other tabs, like the peptide item is going to bring you for more detail, like for the spectrum and the fragmentation patterns for the peptide. So, and LCMS is what you already saw. I'm not going to explain more for the in the other tab, because like this is something that we already covered. Okay, but I'm going to want to show you is the difference of each tab. So by going to each step, you possibly increase your protein identification, because if there's a post-translational modification on your peptide, the identification, like de novo will not be able to recognize it, because the database will not be recognized because the mass change between peptide, like here, will not be the same. But if you add one onto your sequence, then it can actually calculate that change. So by each step, you are like increase like the better of recognizing more peptide to your protein. So here is another next step of PTM. Usually on a normal runs, like a regular run, you're searching the PTM that is like 330 built-in PTM that intakes. For me, for my experiments, I'm only looking at some iodinated peptide because I have lost. So just to like reduce the analysis time on peaks, I only look for those for now. Later we might want to look for more later, but that is how we are going to like that. Like I want to show you the difference in between steps. And since it's pulling up again, I'm going to pause the video for a bit, just to save your time. Okay, so it pull out. So you can see it's pretty much identical output from what we have before. But here is what I want to show you, just looking at the same protein that I want to look at. So just comparing two tabs for the same protein. Just going back to the protein. Okay, here. Just looking at the PTM session here, that it only looks for three PTM. And then there's let's just look at here, there's 110 peptide. And when you go into the PTMs, we actually increase the peptide by 33. There's 133 peptide, and we have like this much of difference modification we're looking at. So you can see like there's a lot more colorful in your protein coverage map. And if you want to, you can export that too, but that is not at the scope of like this video. Okay, here we're just going to like understand the data. And if you have like more detailed questions, just ask like your lab mate, grad students in the lab, and just like they know better. Okay, so like you can see, it's a lot more colorful than it was right here, and we also have more peptide. Okay, so that is like one difference. That's a need for that. And the next step is like spider. I think I have this one pulled out already because I look at that already. And then transferring like is what I want to show this one, and this one like transferring is actually the other one I'm looking at. I need to understand. So here, like let's say like by each one, like I just mentioned that by each step we increase the peptide numbers. And like for spider, we look at point mutations, and then just looking at here, like now we have 199 peptide right now. So what are the difference between these steps? Like you can see on this list, we have like tons of point mutation here, and it is indicated by a box. So here it's saying like a cysteine is mutated to a alanine here. That is the mass change. So that is how you can like see that, okay, we have some of point mutation here. So not necessarily important for your analysis, but it gives you more peptide ID, like depends on what you're going to do. So always remember to check the spider box. Okay, the last one is label-free analysis. That is this one, like that is different from all the three that I mentioned. It's like actually doing a relative quantification for your analysis. And then you can, here is a volcano plot that tells you how significant of your change on this axis and the ratio. So normally the filter is like 20 significance, and fold chains have to be 2. I already turned off all my filter here just for me to understand, like, because I want the most of data. The advantage of label free is it do the normalization for you. So all the time, if you want to export your data to do further analysis, you go to label free because it normalizes to the TIC in between run for you. So you get a sense of the retention time. So here, if you go to like just going back to a transferring, actually I might want to do this. Anyway, this like so in the label free, it can like for the same peptide across the samples, it tells you like the signal intensity in between each one. I think that's like is one way for you to check retention time too, and the area depends on your like what you're doing. And for voting assays, these are really crucial for us. Just an example of like, okay, that's not good. Pretty one. Anyway, let's see. Let me find something that looks good for voting analysis. Actually, they did not look that good. Well, this is better, but you know what? Like the best is the one I just clicked, the first one that we went through. Have a really pretty Gaussian curve and they overlap, something like that. Okay. So I think at this point I tell you most of the stuff about like looking at the data. So if you have further questions for your application, especially if you're part of talk to your grad student, but this introduction should be good for you to start your lab 6 and then like the training, like the mock data analysis practice. And it will be a good practice for you when you're doing your, like you get your data for your 5B and 5C that you can find out something interesting to learn from. Okay, hope you learn a lot from this and let us know if you have more questions.